Good morning, good afternoon, and good evening. Welcome to the ACOD Reader's Briefing, the podcast that turns a full issue of a major magazine into a focused 30 to 60-minute audio briefing. Today, we are bringing you a reader's guide to the 30th of July, 2026 issue of Science. The cover presents a three-dimensional view of North, Central, and South America. Fossil evidence from Mexican mammals suggests that this region served as a biodiversity holding area for around 10 million years before the Great American Biotic Interchange. Rather than allowing species to move freely and rapidly between the continents, the region slowed, filtered, and redirected their movement both northward and southward. The cover also draws attention to several other major stories. These include shortages of a long-acting drug for preventing human immunodeficiency virus infection, neuromodulation as a treatment for dangerous blood pressure crises after spinal cord injury, and two possible routes towards scalable Perovskite solar modules. This issue revolves around 6 major questions. First, when 2 doses of lenacapavir each year can almost completely prevent human immunodeficiency virus infection, will drug supplies, reductions in international aid, and missing community services determine whether the treatment reaches the people who need it most? Second, when every additional day above 30 degrees Celsius during rice reproduction reduces crop yields, how much food security risk are global agricultural models still failing to capture? Third, can calcium isotopes preserved in shells from more than 100 million years ago reveal how volcanic carbon dioxide, ocean acidification, reduced calcification, and selective extinction became parts of one connected process? Fourth, can a non-fullerene polymer electron transport layer combined with plasma surface engineering solve the challenges of efficiency, heat stability, and large area manufacturing at the same time? Fifth, as brain stimulation moves away from deep implants and toward functional circuits closer to the surface, and as spinal stimulation retrains the neural circuit responsible for deadly blood pressure crises, how might neuromodulation transform clinical treatment? And sixth, when artificial intelligence unicorns produce few scientific papers, release little code, and leave behind limited, independently verifiable evidence, what knowledge gaps will confront researchers, regulators, and the wider public? Taken together, the articles in this issue examine how scientific breakthroughs move from discovery into real-world systems. A medical treatment may be highly effective, but access still depends on manufacturing, funding, and community delivery. A climate model may be sophisticated, but it can still underestimate the biological damage caused by extreme heat. A new energy technology may perform well in the laboratory, but it must also remain stable, scalable, and affordable. Across medicine, agriculture, climate science, neuroscience, artificial intelligence, and clean energy, the central question is not simply whether a technology works. The deeper question is whether science can understand the full system around it, including the environmental pressures, institutional weaknesses, production limits, and evidence gaps that determine its real impact. Next, let us turn to the individual article briefings in today's issue.
Section 1. Climate, Oceans, and Ecological Resilience. 1.1. Underestimated heat risk to rice yield. Rice feeds more than half of the world's population. Researchers combined field warming experiments from around the world and found that, during reproductive growth, each additional day above 30 degrees Celsius reduced yield by about 1.1 to 1.8%, far more than global crop models predicted. After recalibration, each 1 degree Celsius of global warming was projected to reduce total rice production by about 8%, with South and Southeast Asia facing the greatest losses. The work shifts warning systems from whole season averages to the narrow heading and flowering window, linking daily weather, crop calendars, and harvest outcomes. 1.2. Witness to ocean acidification. Foraminifera record the carbon dioxide level and carbonate saturation of seawater in calcium isotope ratios preserved in their calcium carbonate shells. About 113 million years ago, rising atmospheric carbon dioxide and ocean acidification near the end of the Aptian may have caused one of the largest extinctions of planktonic Foraminifera in the Cretaceous. The isotope record rose as abundance, average shell size, and calcification declined. The replacement of large, thick-shelled forms by small, thin-shelled forms points directly to reduced carbonate saturation and to broader changes in pelagic carbonate production and the carbon cycle. 1.3. Calcium isotopes link ocean acidification to Aptian to Albian foraminiferal extinctions. The second largest extinction in the evolutionary history of planktonic foraminifera occurred at the Aptian to Albian boundary. Researchers measured calcium isotope records from planktonic and benthic foraminifera, bulk carbonate, and authigenic calcite in the South Atlantic. The planktonic values rose sharply alongside slower calcification, smaller bodies, lower diversity, and thinner shells. Tests of secondary calcite, calcium residence time, and sample preservation ruled out major alternatives, supporting ocean acidification as the driver of this selective extinction. The surface ocean was hit first, and temporary buffering at depth did not mean the wider marine system escaped loss.
1.4. Beyond the Scales, Marine Archaeogenomics for the Conservation and Restoration of Fisheries. Marine conservation is essential for food security and climate resilience, but many management programs rely on ecological baselines that were already altered by centuries of human use. Marine archaeogenomics combines ancient deoxyribonucleic acid with whole genome sequencing to reconstruct past population size, migration, trade, fishing pressure, and environmental response. Marine fish biomass is estimated to have fallen by 38% during the industrial era, and a population that has recovered in number may still have lost historic genetic connections and spawning structure. Archaeological samples can restore references for effective population size, genetic diversity, and former spawning grounds, allowing recovery to be judged by abundance, diversity, and ecological connectivity together. 1.5. China's cordgrass removal increases erosion. China began a national program to remove invasive cordgrass in 2023 and had cleared about 97,300 hectares by the end of 2025. Yet rapid vegetation removal weakened the biogeomorphic buffer that protected coastlines from waves and storms. After clearance, shoreline retreat in the Yellow River Delta rose from 22.1 meters per year to about 144 meters per year, while exposed tidal flats in northern Hangzhou Bay experienced turbidity more than twice that of native vegetation restoration areas and lost more than half a meter of elevation. The authors call for native vegetation, or nature-based buffers, to be established first, clearance to proceed in stages, and shoreline, position, elevation, turbidity, and vegetation cover to be measured so that removal is not mistaken for restoration. 1.6. Repurposing lysosomes for photosynthesis. Reef-building corals and other cnidarians keep photosynthetic algae inside their cells. Researchers found that the compartment surrounding the algae recruits lysosomal proteins and nutrient transporters to create an acidic environment rich in macromolecules, ions, and metabolites. The bicarbonate transporter, SLC26A1, was essential in both sea anemones and reef-building corals, delivering inorganic carbon around the algae for photosynthesis. The work shows that stable symbiosis depends on the host actively maintaining acidity, ions, and inorganic carbon supply.
Section 2. Energy Materials and Chemical Manufacturing. 2.1. N-type polymer layers enable efficient, scalable, and thermally stable perovskite solar modules. Inverted perovskite solar cells commonly use fullerene electron transport layers that are costly, hard to scale, and unstable. The team designed a non-fullerene perenylene bisemide conjugated polymer that improved electron transport, film uniformity, and interfacial binding at the same time. Small cells reach 27.8% power conversion efficiency, while a 625 square centimeter module reached 22.5%. A 30 by 30 centimeter module operated outdoors in Hong Kong for more than 8 months and retained about 96.9% of its initial efficiency. The study placed efficiency, coatability, heat resistance, and module size within one material design. 2.2. Plasma surface engineering for efficient and stable perovskite solar cells and modules. Much of the instability in perovskite solar cells begins with defects that form and evolve at the interface. The researchers first used argon plasma to lightly etch the surface, then reacted fluorine radicals with undercoordinated lead to form a 4 to 7 nanometer perovskite and lead fluoride interface, including inside laser-scribed interconnection regions. Defect density fell by about 1 order of magnitude, small cells reached 27.2% efficiency, and a 100 square centimeter module achieved 23.5% certified efficiency. After 1,600 hours at 65 degrees Celsius under continuous light, the module retained 99.3% of its initial performance.
2.3. Catalytic Appel fluorination of alcohols with potassium fluoride. Conventional Appel halogenation is poorly suited to alkyl fluorides because fluoride becomes trapped by phosphorus reagents. The team introduced a bench-stable neopentoxyphosphonium salt that allowed many alcohols to react with inexpensive potassium fluoride under hydrogen bond phase transfer catalysis, including enantioselective fluorination. The phosphonium salt was prepared on a 148-gram scale, several substrates were fluorinated on the gram scale, and triphenylphosphine oxide and the biurea catalyst could be recovered. 2.4. Stealthy shells enable superlattice assembly. Colloidal particles normally favor dense packing. The researchers first grew complete cubic silver shells around gold nanoparticles, allowing face-to-face assembly into a low-density cubic superlattice as the solvent evaporated, and then selectively etched away the silver. Van der Waals attraction between the gold cores preserved the cubic topology after the shells disappeared. This shape, first, remove later strategy, locked the material into an open structure that would otherwise be difficult to reach.
Section 3. Public Health and Clinical Intervention. 3.1. A Rocky Rollout. Lenacapavir is given as 2 injections, and trials in 2024 showed that 1 dose could provide 6 months of protection and almost completely prevent human immunodeficiency virus infection. A South African model suggested that if about half of the highest risk groups used it by 2030, the country could effectively end its epidemic by 2039, while increasing human immunodeficiency virus and acquired immunodeficiency syndrome spending by only 9%. Supply, however, fell far below demand. By mid-June, South Africa had received only 38,000 doses, and Zambia only 13,000, and some clinics began turning people away within days. Low-cost generics were not expected before 2027, while cuts to United States aid weakened counseling, community staff, and services for marginalized groups. Implementation studies are testing mobile clinics, peer navigation, pharmacies, and schools. But the breakthrough still depends on money, manufacturing, reliable delivery, and sustained uptake. 3.2. Surfacing brain stimulation. Deep brain stimulation can substantially improve Parkinson's disease, but traditional treatment has relied on fixed anatomical targets. Using data from 863 patients, researchers mapped stable connections between the motor cortex and the subthalamic nucleus, globus pallidus, putamen, thalamus, and substance tigra, identifying a somatocognitive action network circuit for Parkinson's disease. Excess connectivity in this circuit tracked both motor and non-motor symptoms. By locating a surface entry point in each person's functional network, non-invasive stimulation may reach the same system from a safer position. The transferable method is to discover the circuit, locate the individual entry point, test it non-invasively, and only then decide on long-term stimulation. 3.3. Rewiring the paralyzed body's false alarm. After spinal cord injury, ordinary signals such as a full bladder, bowel stimulation, or pressure on the skin can be amplified into autonomic dysreflexia, a sudden hypertensive crisis that may cause stroke or death. Whole spinal cord activity mapping and single-cell transcriptomics identified excitatory neurons expressing the transcription factor VSX2 in lower thoracic and lumbosacral regions. Removing those cells prevented the typical blood pressure crisis in chronically injured mice. Repeated epidural electrical stimulation then used the adult spinal cord's plasticity to retrain the maladaptive circuit, shifting care from emergency response after an attack toward reducing the probability that the crisis occurs at all. 3.4. Gene editing death prompts probe. Shanghai Jiaotong University School of Medicine announced a comprehensive investigation into the death of a 6-year-old girl after experimental gene therapy. She was the only participant and was intended to be the first person to receive gene editing directed into the brain. 7 days after treatment, she developed a severe immune reaction and died. The death did not appear in the public trial record or in the later preclinical paper. The investigation will examine the scientific basis, approvals, the family's $860,000 payment, serious adverse event reporting, and disclosure in the publication.
3.5. Immune markers to flag risk. Researchers studied 109 people with relapsed or refractory multiple myeloma who received cilta-cod-tago blue cell chimeric antigen receptor T-cell therapy. Pre-treatment disease features did not predict neurotoxicity. The risk signal appeared after infusion, when higher peak lymphocyte counts, especially stronger expansion of CD4-positive therapeutic cells, were associated with later neurological complications. Peak absolute lymphocyte count is available through routine blood testing and may allow clinical teams to intensify monitoring before severe symptoms appear. 3.6. Making antimicrobial knowledge contagious. In a public school in Kathmandu, 8th grade students used culture plates to observe bacteria from their hands, tongues, and classroom surfaces, then discussed how antibiotics treat bacterial infections and how misuse promotes resistance. The lesson did not present antibiotics as drugs to avoid. Instead, it helps students distinguish bacterial from viral illness, complete a correct course, and recognize unnecessary use. The author hoped students would carry this evidence-based understanding back to their families and communities. 3.7. Variant variety. Chimeric antigen receptor T-cells are made from a patient's own T-cells and therefore carry inherited germline variants. Researchers linked clinical outcomes in 2 lymphoma trials with germline sequencing and found that very few variants were associated with adverse outcomes. A small number tracked more severe toxicity, stronger inflammatory cytokine production, and macrophage activation, whereas others tracked lower toxicity or greater therapeutic cell expansion. These associations remain candidates for further validation and are not yet a genetic screening rule. 3.8. Expanding peptide access. An advisory committee to the United States Food and Drug Administration recommended allowing compounding pharmacies to manufacture and sell 6 peptide products. Supporters argued that regulated pharmacies could reduce a rapidly expanding online gray market, while agency scientists noted that several candidates lacked safety, efficacy, and human exposure data matched to the proposed route of administration. The committee rejected imetiltide because the evidence was weak. The final decision still has to balance a safer regulated channel against the risk of giving medical legitimacy to poorly supported products.
Section 4. Research Governance, Artificial Intelligence, and Evidence Policy. 4.1. Artificial intelligence's top startups are barely publishing their research. A preprint examined 317 artificial intelligence unicorn companies that existed between 1998 and 2025. The final dataset contained 2,700 publications, yet more than half of the firms had never produced a paper with a company researcher as first or last author, and their combined output in 2025 amounted to only 1 in every 1,000 artificial intelligence papers. Scientific influence was also concentrated in a small number of firms and repeat authors. Papers are not the only form of disclosure, but companies that claim to be transforming science and society still need to release methods, code, data, or model weights that outsiders can independently test. 4.2. Golden Age Report is a missed opportunity, critics say. The White House report Science, A New Golden Age, proposed sending less federal money to major research universities, shifting support from investigator-led small projects toward large teams working on grand challenges, and using alternatives to peer review. Many scientists found the title difficult to reconcile with thousands of grant terminations, proposed agency budget cuts, and greater political oversight. The report included ideas worth testing, such as re-granting and a reviewer's golden ticket, but it did not explain why one externally imposed priority corrupts science while another priority set by the administration repairs it. The real test will be whether budgets remain stable, professional capacity is retained, and basic discoveries can become domestic industries without being sold too early. 4.3. Rigor is not readiness. The Policy Evidence Readiness Levels Framework for Evidence-Based Policy. Social and behavioral science adopted more rigorous trials, yet interventions that worked in early studies often produced small, zero, or negative effects when expanded. Policy evidence readiness levels divide evidence maturity into 4 stages and state what each stage can support, from an initial insight through controlled efficacy and real-world effectiveness to systematic synthesis. The framework asks policy action to match evidence maturity. An early result can justify another test, but it should not be presented as proof of universal effectiveness. 4.4. The National Institutes of Health is again terminating politically sensitive grants. The United States National Institutes of Health resumed terminating grants on health disparities and other politically sensitive topics, including a 5-year study of how racial discrimination changes brain activity. The new termination letter argued that the measure of discrimination was subjective and that studying only Black participants limited comparison. A court had already ruled more than 1,700 earlier terminations unlawful, and the new procedure now faces another ruling that agencies cannot cancel existing awards merely because priorities later changed. Participants already recruited, trained staff, and established data systems may not be easy to restart.
4.5. The United States unveils the first Genesis mission grants for artificial intelligence in research. The United States Department of Energy announced the first 278 Genesis mission awards for artificial intelligence in research, covering nuclear reactor design, soil microbes, critical minerals, and other problems. Teams had only 6 weeks to assemble cross-institutional applications, and most winners entered a 9-month first phase competition. The initial success rate was below 5.6%, prompting concern that money had been diverted from existing programs and could squeeze theory and experimental construction. The next evaluation will need to separate real scientific progress from short-term demonstrations. 4.6. Privacy-preserving tools banned in the census. The United States Department of Commerce banned the Census Bureau and Bureau of Economic Analysis from using modern privacy methods such as noise infusion and required them to rely on coarsening and suppression. Differential privacy can formally limit how much one person's information affects a published result, although it can also distort small area data. The directive did not continue the technical debate over privacy and accuracy. It prohibited the tools outright. Critics warned that this could erase information about small groups, weaken public data, and allow political appointees to dictate statistical methods. 4.7. Dismantling diversity, equity, and inclusion has consequences for all of science, technology, engineering, and mathematics. An executive order declared programs focused on diversity, equity, and inclusion unlawful, and many science, technology, engineering, and mathematics institutions froze, renamed, or canceled related work. A case study argued that the change did more than remove an office name. It altered recruitment, mentoring, training, data collection, and participation. Once the programs were dismantled, institutions also lost the ability to record who was excluded and where people left the pipeline. The consequences may appear years later as a narrower talent pool and less varied research teams. 4.8. Churn, not crisis, for United Kingdom science. Prime Minister Andy Burnham abolished the United Kingdom Department for Science, Innovation and Technology and folded it into a Business, Innovation, Science and Trade Ministry. The editorial argued that Britain had reorganized the machinery of science policy without undermining its foundations because the £86 billion research and development settlement and United Kingdom research and innovation remained in place. The deeper weakness was late-stage capital. Many university spin-outs were sold when they needed large investments. The reshuffle will succeed only if it turns discovery into prosperity without crowding out the science that creates discovery.
Section 5. Biomedical Mechanisms and Therapeutic Technologies. 5.1. Spatiotemporal multiomics uncover tumor ecosystem dynamics during metastatic colonization. Metastases causes most cancer deaths, yet the earliest stages after disseminated tumor cells reach a second organ remain poorly understood. Researchers combined high-resolution spatial transcriptomics, single-cell ribonucleic acid sequencing, and chromatin accessibility analysis across 9 stages of lung colonization in mice, then validated the pattern in human samples. Natural killer cells and neutrophils removed most cells within the first 12 hours, while a small surviving population entered a transient state marked by high phosphoglycerate dehydrogenase, low proliferation, and sparse immune contact. Metastases emerged as a sequence of ecological transitions that can be intercepted at different times, with early therapy helping immune cells reach survivors and later therapy blocking growth support from macrophages. 5.2. Enabling sustainable supply of the essential cancer medicines etoposide and tenopicide in yeast. Etoposide and tenopicide treat millions of cancer patients each year, but their supply has long depended on the endangered Himalayan mayapple and on more than 10 stereoselective chemical steps. The researchers engineered yeast to make, from glucose, a key intermediate that already contains the aryl tetraline scaffold, the correct stereochemistry, and the carbon-4 glycosidic bond. A single mild chemical step can then produce either medicine. Modular engineering controlled precursors, coupling, oxidation, and glycosylation while reducing dependence on destructive harvesting, climate, and the plant's long growth cycle. Industrial production still has to improve titer, medium cost, oxygen transfer, purification, and genetic stability. 5.3. Atlas of Lysosomal Aging reveals a metabolite signature shared with lysosomal storage disorders. Lysosomes break down proteins, lipids, and other biological molecules, and also participate in nutrient sensing and metabolic signaling. Researchers use tagged and tag-free methods to isolate intact lysosomes rapidly, then combined metabolomics and lipidomics to build an aging atlas across tissues from mice, rats, and rhesus macaques. Glycerophosphodiesters and cysteine accumulated with age in several organs and species and overlapped with signatures of childhood lysosomal storage disorders. Calorie restriction reduced the buildup in only some tissues, showing plasticity without proving that the organs had become younger. The tag-free method can also reopen frozen archived samples and provide a common reference for testing interventions. 5.4. Signatures of aging and disease in a single organelle. This perspective placed the lysosomal aging atlas between normal aging and childhood lysosomal storage disorders. The appearance of the same metabolites from different starting points suggests convergence on a related organelle imbalance. Glycerophosphodiesters and cysteine may help build an organelle aging clock, but predicting age is not the same as identifying a cause of aging. Only experiments that change these metabolites and restore function can show whether they drive decline or merely record it.
5.5. Follow the calcium. Adult heart muscle cells do not divide effectively, so damaged myocardium cannot fully repair itself. In cardiomyocytes derived from human-induced pluripotent stem cells, calcium transients and contraction fell temporarily as cells entered mitosis, reached their lowest point in metaphase, and recovered after division. The sarcoplasmic reticulum calcium pump, SERCA2, accumulated at the spindle poles, creating a quieter mechanical window for chromosome alignment and separation. Any regenerative treatment will have to control the timing and location precisely, so that a small number of cells divide and then recover mature contraction. 5.6. X chromosome inactivation draws long interspersed element 1 mutagenesis to the human X chromosome. About 30% of the human X chromosome consists of long interspersed element 1 sequences, roughly twice the average on autosomes. Long-read methylome sequencing showed that new insertions preferentially entered the late replicating inactive X chromosome, meaning the enrichment first reflected integration preference rather than later selection. The inactive X can silence a mutation temporarily while allowing it to enter the germline and pass to a male child, where the damaged gene becomes the only copy. The mechanism connects replication timing, chromosome state, and X-linked disease risk. 5.7. Ribonucleic acid editing for stem cell health. The ribonucleic acid editing enzyme ADR limits proliferation in fruit fly intestinal stem cells, but its level falls with aging and after gut injury. Young flies that lost the enzyme developed excessive stem cell proliferation, impaired intestinal homeostasis, and shorter lives, while restoring expression in old flies improved gut function and extended life. The work linked reduced editing to the pumilio-regulatory axis and stronger mitogen-activated protein kinase signaling. Similar changes were also observed in the aging human colon and during mouse intestinal regeneration. 5.8. Why the X chromosome is rich in long interspersed element 1 mobile elements. Long interspersed element 1 copies its own ribonucleic acid into deoxyribonucleic acid and inserts it elsewhere in the genome. The new study showed that, before selection acts, it enters the late replicating inactive X chromosome 2 to 3 times more often than the active X or autosomes. This weakens the idea that the element accumulated mainly because it helps spread X chromosome inactivation, although accumulated copies may still reinforce silencing later.
5.9. The molecular architecture of mammalian vitreous body collagen fibrils. Researchers used relatively uniform collagen fibrils from the pig vitreous body in cryo-electron microscopy to resolve their native molecular structure. Type 2 collagen ran in 2 opposing orientations. Type 9 collagen and optissin occupied the surface, and sugar chains appeared at conserved sites that controlled packing density and hydration. This architecture lets the fibrils support the interior of the eye without forming large bundles that scatter light, providing a framework for understanding collagen disease and designing artificial vitreous arterioles.
5.10. Sugar-selective neurons. Researchers compared how equal calories of glucose and fructose suppressed agouti-related protein neurons in the mouse hypothalamus. Fructose activated a distinct gut-to-brain pathway, inhibited the neurons less strongly than glucose, and altered food preference. When vagal signaling from the gut to the brain was interrupted, the fructose effect disappeared, while the glucose effect remained. The neurons therefore detect the identity of a nutrient, not only its calories.
5.11. An adapter for feedback regulation of heme biosynthesis by a mitochondrial protease. When heme is abundant, the rate-limiting enzyme 5-aminolevulinate synthase is degraded by a mitochondrial protease complex. The study found that the adapter POLDIP binds the heme-occupied enzyme and delivers it to CLPX and CLPP. Heme is both the metabolic product and the molecular glue that joins substrate to adapter. This feedback mechanism explains how mutations linked to porphyria can cause excessive precursor production and identifies a possible point for intervention. 5.12. Deoxyribonucleic acid polymerization activates ribonucleic acid cleavage of a reverse transcriptase-like antiviral enzyme. The defense-associated reverse transcriptase, DRT4, is not a conventional reverse transcriptase. One protein performs deoxyribonucleic acid polymerization, exonuclease activity, and ribonucleic acid cleavage. Phage infection raises intracellular nucleotide levels and supplies a single-stranded deoxyribonucleic acid binding protein, shifting the balance from degradation toward polymerization and then activating ribonucleic acid cleavage. The host enters dormancy and blocks the virus, placing infection sensing and the execution response in the same protein.
Section 6. Physics, Astronomy, and Research Infrastructure. 6.1. Iconic telescope to shutter amid budget cuts. The United Kingdom Science and Technology Facilities Council plans to end core funding for the radio network that includes Jodrell Bank Observatory and the 69-year-old Lovell Telescope, which may close within 2 years. The loss would affect more than one instrument. It would disrupt 7 antenna coordination, technical training, and long observational time series. The same round of cuts also threatens dark matter searches, solar monitoring, and exoplanet surveys, and scientists and research unions have pledged to oppose the decisions. 6.2. Will Earth survive the Sun's expansion? After the Sun exhausts hydrogen in its core, it will expand through red giant and asymptotic giant branch stages, potentially beyond Earth's present orbit. Stellar mass loss pushes planetary orbits outward, while tides pull them inward. Updated models predict that Mercury and Venus will be engulfed and Mars will survive. Earth may move outward far enough to escape, but uncertainty in asymptotic giant branch mass loss still leaves its fate unresolved. Orbital survival would not mean that the surface remained habitable.
6.3. Toward an exact quantum many-body treatment of Kondo correlation in magnetic impurities. The Kondo effect arises from many-body interactions between a localized electron on a magnetic impurity and itinerant electrons in a metallic host, and its characteristic temperature is extremely sensitive to model parameters. The researchers began directly from a material-specific ab initio many-electron Hamiltonian and systematically expanded the orbital space around the impurity for 7 transition metal atoms embedded in copper. Without fitting to experiment, the method reproduced subtle trends in Kondo temperature and allowed its error to be checked by enlarging the orbital and solution spaces. 6.4. How black holes get hitched. Gravitational wave observatories have recorded more than 300 black hole mergers. In population studies now indicate at least 3 formation routes. Low mass pairs are consistent with the evolution of field binaries. Intermediate mass pairs look more like dynamical pairing in dense environments. And the most massive systems may contain a black hole created in an earlier merger. The evidence still comes from a limited event sample, so additional data may split the 3 groups further or reveal new formation environments.
Section 7. Scientific Culture, Education, and Research Integrity. 7.1. The Perils of Fluency. The author once mistook shared terminology and smooth discussion for shared understanding. Only when an interdisciplinary team converted its conversation into a concrete research plan did members realize they had been using the same words for different ideas. She began allowing pauses, uncertainty, and unfinished explanations to last longer and used restatement and concrete examples to test comprehension. Fluency can feel like mastery, but better tests include delayed recall, transfer to a new problem, and the ability to explain an error. 7.2. Editor's Note on Ebola Report Images. Science found that several supplementary images in a 2014 Ebola hemorrhagic fever study had used incorrect or repeated source material, and some original data could no longer be recovered. Survival, viral load, coagulation, and other evidence still supported the main conclusions, so the journal did not retract the paper, although support for a specific tissue inflammation claim was weakened. The episode shows that long-term preservation of raw data and clear records of image provenance are basic conditions for later verification. 7.3. Editorial Expression of Concern. A paper on hafnium oxide interfaces in perovskite solar cells reported high efficiency and about 5,000 hours of operational stability. But readers noted that the main stability curve appeared to contain identical data segments at different times. Science issued an editorial expression of concern and asked the authors for the raw time series, instrument exports, and figure generation records. Until the investigation is complete, the long-term stability claim should be used cautiously. 7.4. The Optimism of Doubt. Stuart Firestein's book argues that one of science's greatest gifts is not a collection of certain answers, but a structured relationship with uncertainty that can continue generating knowledge. Moving from Copernicus and Darwin to thermodynamics and chaos theory, the book treats uncertainty as a permanent feature of reality. The review adds an important boundary. Productive doubt must remain testable, accept failure, and permit revision, rather than becoming a license to manufacture confusion.
Section 8. Evolution, Ancient Deoxyribonucleic Acid, and the History of Life on Earth. 8.1. The genomic identity of early smallpox in South America. Researchers recovered the earliest ancient smallpox genomes from the Americas in the bones of 2 people from northern Chile who lived during the transition from Inca to colonial rule, approximately between 1492 and 1631. The 2 sequences were 99.912% similar, suggesting the individuals died in the same outbreak. The virus belonged to a lineage related to ancient European smallpox, providing direct molecular evidence that the disease entered an indigenous community after colonial contact. 8.2. When mammals crossed between continents. The Isthmus of Panama connected North and South America about 2.8 million years ago, yet South America today contains far more mammals of North American origin than North America contains mammals of South American origin. New Mexican fossils support a 2-way biogeographic holding area in which species paused, adapted, and diversified at low latitudes before a later migration pulse. Selective losses during the late Pleistocene amplified the imbalance. The same pattern suggests that modern invaders may remain quiet for a long time and then expand rapidly. 8.3. Ancient deoxyribonucleic acid shows how smallpox reached the Americas. Researchers identified smallpox virus in the bones of 2 naturally mummified people from northern Chile, filling a long-standing gap in the molecular record of colonial America. The hosts were indigenous Americans, while the virus belonged to an ancient European lineage, and the outbreak dated to after European arrival. The report also covered debate over whether smallpox evolution slowed during the colonial period, but researchers agreed that even a less virulent strain could have had an extreme effect on populations with no previous exposure.
8.4. A 10-million-year biogeographic holding pen primed the Great American Biotic Interchange. Researchers expanded the record of Neogene mammal genera at Mexican fossil sites from 919 to 1,724 and combined it with data from South America and temperate North America. The analysis proposed a 3-stage interchange lasting about 10 million years. Mexico first accumulated northern species, then created a latitudinal delay, and finally released large migration pulses after the land bridge became passable. Mexico was therefore not a passive corridor, but a biogeographic pacemaker that controlled the tempo of exchange. 8.5. Difficult births. Researchers found that applying measurements designed for human obstetrics to non-human primates overestimated the space in their birth canals. Species-specific measurements of adult female pelvis and newborn skulls from 29 primate species showed that some small monkeys and prosimians had a tighter fit than great apes. Difficult birth is therefore not a uniquely human condition, but an evolutionary trade-off involving fetal size, pelvic shape, maternal body size, locomotion, and fetal presentation. 8.6. Supergene control of chiral development in mirror image flowers. The mirror image flowers of the South African butterfly lily occur in stable left and right forms. Developmental imaging, biomechanics, genomics, and natural mutants showed that intrinsic organ twisting and gravity responses amplify the bend, while a supergene containing auxin-related and microribonucleic acid components coordinates the directions of the style and anthers. Closely linked developmental genes are inherited as one unit, keeping pollen placement, stigma position, and pollination function aligned. 8.7. A gene helps windswept lizards get a grip. After 2 hurricanes crossed the Turks and Caicos Islands in 2017, surviving anole populations had longer forelimbs, shorter hind limbs, and larger toe pads. By comparing genomes before and after the storms, researchers identified the candidate gene HS6ST1 and knocked it out in embryos of a related species. Loss of the gene shortened the 4th toe of the hindfoot and increased grip in a small storm simulation. The result shows how a few extreme events can drive pulses of change in both form and gene frequency.
All right, we are now ready to enter today's main program. Before we do, please like, save, and share this episode so that other people who need it can also see today's content and become better versions of themselves. Thank you. You can now use the timeline in the video description or the pinned comment to find the articles that interest you and listen to them in depth.